Well, hi, good afternoon. I would like to talk today about one of my artificial intelligence projects in the laboratory. We used to treat 10 years ago, we used to treat cancer as one size fits all, but we now know that every patient has his own cancer mutation profile. You can see that this is an example of patient of mine, and she has mutation in three different genes, several mutation, and we can also prescribe some proposed medication. This is what about precision medicine is. The problem is that this is the tip of the iceberg. They report on the 3 or 5 mutations that are surely pathogenic. They are surely participate in the process of. Thank you. Is it better? Okay. Okay. Actually, I can do without it. They need it for this one. Okay. Okay. No problem. So this is actually the tip of the iceberg, because we know that there are much more mutations that are found in the tumor, and they are defined as variant of unknown significance. So you can see here a lot of mutation. This is and we don't know what to do with them. They write them in the appendix. And why is this important? We divide the mutation into two different types of mutation. There are somatic mutations that exist in the tumor itself, but not in the body, in all the cells of the patients. And there are germline mutations. So for the somatic mutation, around 80% of all mutations that we find in cancer genes are somatic. And this is important because we now know from, for example, one study of Razel Kurzrup in 2019, that if we treat more than we target by drugs more than 50% of the driver mutation of a tumor, then the survival is much better. So maybe we treat two mutations that we know about, but maybe those variant of unknown significance hide 2 or 3 more pathogenic mutations. We are just not aware of it because it's inside the forest of 30 different mutations. And not less important is the germline mutation. There are patients, 13% of cancer patients, sporadic cancer patients, carry an inheritable mutation, but 47 of them are carriers of variant of unknown significance in cancer genes, and we don't know to tell them whether they are in risk for additional cancer or their relatives are in risk for additional cancer. And I would like to give an example of how we try to address this problem. So cancer is caused by genetic mutations, and one of the most important genes in cancer is P53, which prevents mutation. And if there are too many mutations, it causes apoptosis, which is program death, program suicide for the cells, which prevent cancer. The cancer is smart, so in the fight between P53 and the cancer likes to select mutation that disrupt P53, and then the cancer wins. Indeed, 40 to 50% of tumor harbor P53 mutation, and I work with Professor Thierry Soucy, who is collaborating one of the leaders in the research of this gene. And some of the mutations are heritable and confer cancer risk. This is the Low-Frumeni syndrome, which is risk of 50% chance of cancer before the age of 30. What is the problem? You can see here the landscape of different mutation in cancer in this gene. This is the position. You can see that there are some hotspots that recurred many times in cancer. They are of course pathogenic, but many of them were mentioned in this database of 10,000 patients only once or twice. So is this a chance by chance mutation, a neutral mutation, or a pathogenic mutation? And if it's found in a patient or inheritable mutation in the family, we don't know to tell the family anything. And importantly, most of the possible missense mutation in this gene are variant of unknown significance, which we don't know to tell the patient. So what we do, we selected to use machine learning. In machine learning, we define a group of mutations that we are almost sure that they are cancer causing, another group of mutations that we are almost sure that they are neutral, not cancer causing. We collect a lot of information about them, and then we ask the computer to create a model to discern between these two groups, and then this model can be generalized to all other mutations that we don't know about. Now the problem is, and this is what important for me to show, the interface between the artificial intelligence and biology. Okay, so many people are impressed by the fact that in my lab, we know to do artificial intelligence, which is nice for medical people. Okay, but this is not the only issue. The biology is very important. So how we select them? So this one, the pathogenic group. The pathogenic group, actually, we can see the cancer gives us an opportunity, because usually in genetic diseases we have only the mutation of rare genetic diseases, but cancer is prevalent, and mutation that appear in somatic mutation in cancer can actually represent an experiment, so-called experiment of nature, of selecting cancer causing mutations. So if we take 4 different databases and select only the mutation that appear in all of them independently, then we can be quite sure that they are cancer causing. And indeed, if you see here, this group of mutations are more pathogenic, are more disrupting the cells as compared to this group. The next issue, which is even more challenging, is to select the neutral mutation. So we can say, okay, the mutation didn't appear in cancer thus far, that we investigated 20-30 years, is neutral. But who says that in 3 years, when we'll have millions of patients with cancer genomes, we'll not find this mutation, that our algorithm will be wrong. And for this, P53, and this was the idea of my colleague, P53, if you take it, the mutation appearance, the new mutation appearance in history, you can see that after 2008 there is saturation. All the pathogenic mutations were already appeared in cancer. There are no new mutation. This we can assume maybe 95% of confidence that the mutation that didn't appear are neutral. And the data of each mutation consists of many computational scores, many yeast and in vivo cells scores that experimented all possible missense mutation. This is named the saturation mutagenesis. It exists for minority of genes, but for P53 it does exist. And now this is a nice computer game. It's actually true, it's statistics, it's really grounded, well grounded in mathematics, but as a biologist and physician we want more validation. So the first validation is a test set which was left 20% of the mutation that we know that they are positive or negative. They are 96%, 96.5% accuracy of the prediction. In the ClinVar, in the leading cancer interpreting database that is used often by genetic consultation and oncologists, where we have only 190, but out of these 96 again are positive, our prediction is accurate. And a clinical validation, which we took a database TCG of 10,000 cancer patient mutation, we divided the patient into 4 groups. The first group is the patients without P53 mutation in green. The second group is patients with truncating, not missense, not point mutation, but mutation that harm all the genes. We are sure that they are pathogenic. And then all the point mutation, we divided them into the yellow, which we predicted to be non-deleterious, and the blue, which we predicted to be deleterious. And indeed, we can see that the mutation that we predicted to be deleterious go with the truncating, and mutation which we predicted to be non-deleterious are going with wild type. Okay, so this is a very independent validation. And the last validation is experimental. We took 41 mutations that we have interesting biological question about. We did our prediction, we transfected them with my colleague into human cells, and we selected and we showed that this is how a pathogenic mutation behaves. It causes proliferation of cells, and this is how wild type behaves, and we see that our predictions are correct. And we now apply this method to more and more cancer genes using variety of methods. Now here comes biology again. You know that artificial intelligence is not artificial and is not intelligence. The computer does what we ask it for. There is no mystics about it. And we told the computer to divide the mutation into pathogenic and non-pathogenic. So we forced the computer into assumption of dichotomy the computer will not find anything else because we forced it into dichotomy. But is dichotomy correct? So here, and this is a paper under review, so I will just show some hints and I will not elaborate on the method. This is a saturation mutagenesis database that showed the pathogenicity of all mutation, and one is very pathogenic. And this is our score, which is related to evolution and mutation prevalence, but I will not elaborate. I hope it will be accepted soon, and I'll be presented it the next time. And we can see that there is a group of very pathogenic mutation, group of neutral mutation. There are many weak drivers, and this is very important therapeutically. We validated it in survival and so on, but this is very important therapeutically. You can see that the stronger mutation, this is from a cell line encyclopedia, the stronger mutations needs much lower, are more sensitive to the relevant medication, which means that this stratification to weak driver, strong driver, and so on is also important therapeutically. So what we are planning to do it in Hadassah. So in Hadassah we are fortunate to be one of the Sharet Institutes, one of the 4 centers in the world that were selected by Roche company for a very interesting collaboration. This is a collaboration led by Professor Aaron Popovtzer and Professor Eli Pikarsky, and I'm in charge of the scientific and computational parts of this project. And we will have a genomic profile for every cancer patient in Hadassah, 3,000 new patients every year, but we build around them a patient journey, a digital and structured patient journey database that is available also for single patient, for physician as an operative tool, but also for us all the treatment, the pathology, and all the clinical implication of the patient journey. And then we will use, and this is we're starting to do it today, we will use all the big data from pathology, from radiology, from treatments, from genomics, and for clinic, and try to cluster the patients according to sensitivity to drugs, even patients that are far apart from different diseases, from different genes, for different drugs. And then for each new patient, this is the vision which I hope will, for every new patient, we will tell this is your profile, you will need drug B, this is your profile, you will need drug A, and this is your profile, drug A actually will harm you. So this is that we try to use our artificial intelligence and all the tools that I described and more to develop this decision support tool. And these are the brilliant students that I'm fortunate to have, that all of them, aside one who comes from computer science, all of them are medical students, and 5 of them have or will have in 2 years a first degree in computer science or mathematics within with a degree with MD. And I'll be happy to take, this is the work of Gil Ben Cohen and the PhD students, and Kobi Landau, who finished now his master's degree and is doing internship in medicine. So I would be happy to take 1 or 2 questions if we have time.